So my name is Rose, and we have Thomas and Nicole, who are also students here at Cornell. And so first of all, I want to start with an introduction of everyone. So Thomas and Nicole, you can say like what lab you're in, what research you do in that lab, and then briefly describe how you got to Cornell and specifically to that lab. So Thomas, do you want to go first?
Sure. So I'm Thomas Ciavatti, and before coming to Cornell, actually, I studied physics and in particular instrumentation. And I had a very nice thesis project that involved building a microscope, and I had a lot of fun doing it. So I wanted to do more of the same in my PhD, more kind of instrumentation, microscopy type of work. And I wanted to stay away from what I was doing before, which was more like solid state type of samples, and I thought it was in some sense not that interesting. So I wanted to do more biology focused instrumentation. So I came to Cornell for that. And I actually am currently in Warren Zipfel's lab down in the basement of Weil Hall, the imaging core. But that's not where I started. My first group here was I was with Chris Hsu, who his group focuses on 3-photon microscopy, multi-photon microscopy, and deep brain imaging. And I thought the research was very interesting, but the group dynamic really didn't work for me, so I had to switch group. I switched at the beginning of my third year, and I've been with Warren Zipfel ever since, and it's been great. I'm working on a few projects. One of them is building a confocal. So a confocal microscope that's broadband with many wavelengths to look at different molecular tags across the spectrum. I also have a project that involves some biochemistry as well as the microscopy to put together a spatial transcriptomics platform, which basically allows us to take images while at the same time getting the genetic code. So it's really kind of a fascinating project, and other little projects in the lab. It's an imaging course. There's all kinds of instrumentation projects to work on, and I'm happy to answer any questions about all that. Cool.
Okay, great. Nicole, can you give your little spiel?
Okay, so I'm an instrumentation, but from the opposite side that Thomas came from. So I was doing like mostly stem cell research in undergrad, but I was a bioengineering major. And then when I started my PhD, I was like, there's no engineering in stem cells. So I tried to get a little bit more of, I guess, physics and engineering into my PhD work. I actually started my PhD at Johns Hopkins, and kind of similar to Tom's too, group dynamics weren't great. My fiance was also at Cornell, so I transferred. And so it's my second year here, but like fourth year in a PhD program. And I am part of the Schaefer Nishimura lab. So I work on, as similar to Chris Hsu actually, we do multi-photon microscopy. So I do deep brain imaging studying white matter disease. And I'm also working on using 3-photon microscopy to detect blood oxygen concentration. So that's like a little side hopeful project. Did I miss anything I was supposed to cover? Yeah, so happy to answer questions.
Awesome. Okay, well, I am Rose. I am a fourth year in the PhD program, and I work in David Erickson's lab. So he's actually a mechanical engineering professor, but he's in the biomedical engineering field. So we work on infectious disease diagnostics and nutrition diagnostics. So that has been a really hot topic lately. So it's been really interesting for the last year and a half. I came to Cornell because I thought it was a really supportive environment, and I thought that the choice of labs was really good for me. I come from an electrical engineering background with some biomedical engineering in there too, and so coming here was a lot more biology than I was used to, but the transition was relatively easy. I think probably all three of us can talk about like transitions from like one field to another, like slight deviations. because it's really common for biomedical engineers to have to know a lot of these different things. So does anyone have any questions for any of us? You can put them in the chat if you would like.
Hi, I have one for Nicole. So I'm interested in, I'm looking forward to talking to your advisor, but I had a question about, do you all use multi-photon with photoacoustic together in order to enhance, I guess, the resolution and the depth in some type of animal models, or do you guys not focus anything related to that?
We, as far as I know, do not. Doesn't mean we're not open to it. I know Chris and Nozomi are pretty open to looking forward and encouraging students who are interested in certain things to like build whatever they want themselves. So they're pretty supportive of doing the next best thing. So yeah.
Awesome, thank you.
So I do not work on microscopes, but one word that popped out reminds me of like what Steve 80 does. So you could also look at his lab. His lab does some microscopy stuff too. Any other questions? Otherwise, we can go through our list of things that I made a list of what I think we should talk about. Okay, so Thomas, I have a question for you. When you were first choosing a lab, can you describe the timeline and the process?
Well, I chose a lab pretty quickly, maybe a little too quickly, perhaps. But I was just really interested in the research that Chris Xu was doing. This kind of very quantitative approach to instrumentation was very appealing to me. So, you know, I didn't pay that much attention to maybe more personal things that down the road turned out to be more important than I expected. I had always had, to be more explicit, I always had very good interactions with previous advisors or mentors. And so it didn't occur to me that things could go rather poorly, but they did. And so I had to switch groups. I don't know, does that answer, Rose? Maybe you want to instantiate that question?
Yeah, no, that was good. I think choosing a lab is a really like personal process. And specifically at Cornell, I'm not sure how much information you've gotten about this so far, but we have really like informal rotation program, I guess, where you just kind of talk to the advisor and set up meetings with them. You can rotate in one lab, you can rotate in as many labs as you want, specifically during the first semester, and then you make a decision at the end of the first semester. So for me, I actually rotated in the lab that Thomas is in now, and then I rotated in my current lab, and so I chose those two as kind of like the top runners. But I also talked to faculty from neurobiology. I talked to faculty in electrical engineering too, just to see if there was some possibility of me liking a lab in a different department that it was still in the BME field. So like over the course of the first semester, you'll really be trying to figure out what lab you want to go into. Some people already know when they come in, and that's great, but it's a good way to spend the first semester just like figuring out what type of research the labs that you're interested in do. Again, figuring out the group dynamic, making sure it fits for you, and then making sure you get along with the advisor. So you don't come in with a lab specifically, but you do get to choose it relatively quickly and kind of on your own terms, which I think is really interesting. Nicole, so I actually have never met Nicole before. So her story is like brand new for me. But how did you decide to join Chris and Nozomi's lab? First of all, you transferred, and then how did you choose that lab?
So I was in the process of like transitioning to more finding a balance between instrumentation and biology, and Chris and Nozomi's lab, I thought was a really good mix of like, you have people developing the tools and people using the tools. So it was a good place to be where I could constantly learn new things on both sides in biology and instrumentation. I initially wasn't going to join that lab because I didn't want to do animal research. But when I started listing my interest to Jan, he was like, this lab would probably be a good fit. So I talked to them, and I really liked Chris and Nozomi. So yeah, I interviewed with Steve Ady's lab too. And then didn't like it as much. Nothing against Professor Ady. It wasn't as good of a fit. So yeah, and I learned a similar lesson to Thomas at Hopkins, that personality does matter. I know everyone was like saying that when I was joining in Hopkins too, and I was like, it'll be fine. Like I've been able to work just fine with like so many people. Like how bad could it really be? It can be pretty bad. So don't like ignore that part. Yeah. But
So Nicole, what were you looking for in an advisor the second time around?
well, at Hopkins, I learned that I hate being micromanaged. So I was looking for an advisor that did not do that. And Chris and Nozomi are like on the other side of that spectrum, where some people could argue that they should manage a little more closely, but I happen to do well in that environment where I'm able to function off of like my own motivation as opposed to like external pressure. Yeah.
Thomas, do you have any? Oh, sorry. Go ahead.
Sorry. Yeah, just generally as like looking at how people in the lab were, like they were mostly talkative, optimistic. The other lab that I came from, it was like real quiet every day. So yeah.
That's really interesting. Thomas, do you have any comments about what you learned through this process?
I mean, yes, pay attention, really test out the waters of interactions, particularly, how other students perhaps that have gone through the lab have experienced communication or miscommunication with a particular advisor or a particular group. I want to say something else, Rose, and not answer your question anymore.
That's totally fine.
people sometimes ask me, what do you think of Cornell? And there's something really magical about Cornell that I have found, and I want to share it. So before, when I was at Stony Brook, fantastic place, by the way, but I felt like I was in the physics department and there were these four walls and nothing outside of the physics department existed. I mean, with some exceptions, but basically I was just in this department. At Cornell, there's just this incredible interaction between all the departments, and not even just in the College of Engineering, but across the sciences. I took a course, for example, in plant science, not because I'm interested in plants, but because it's a methodology course that had all kinds of things that were relevant to what I was doing. And there's this kind of dynamic. I know lots of people from the music department and humanities, and there's this really kind of university atmosphere that actually is, I think, not that common. And Cornell does a really good job at that. I don't know if Nicole and Rose, you have the same sort of feeling, but that's really something I found really positive about Cornell.
Yeah, I very much agree with that. I can personally attest to a lot of the collaboration that happens, mostly because I literally have to walk like from my building to a different building every day because we're working with either people in biotech building or human ecology, and just being able to use those resources has been really beneficial for my research.
Yeah, there's a lot of collaborations going on, to the point where sometimes people complain about there being too many collaborations. And if I heard that if you like start doing something that some other lab like could have helped you with, they're like, why didn't you ask us for help? It's like, so it's a very positive environment. So
Yeah, so Thomas mentioned classes. Nicole, have you taken any classes here?
yeah.
Yeah. So both Nicole and Thomas, what classes did you take and did you find them beneficial?
I can go first. Okay.
Nicole can go.
I was transitioning in from my neuroscience background was strong, but my physics background was not, and I wanted to learn more about how lasers work and what multiphoton microscopy even is. So I took, Nozomi has a class on, I think like in vivo imaging of living systems or something. I don't remember, but the title exactly. So it kind of goes through different methods that we use to image mice and other animals. And then I also took advanced E&M and nonlinear optics because I wanted to understand the physics of what I was using. So they were all beneficial classes, but that was also by design because I'd already kind of done my master's and like taken a bunch of classes at Hopkins. So I really only had to take like the classes that I needed for my own knowledge.
Yeah, I haven't taken many classes either. There's sort of, I guess, two kinds of classes. I think I would classify them as sort of the useful ones and the not useful ones. And Jeff Moses' nonlinear optics course was really helpful and insightful and applicable. And then I took this E&M course from physics, which was a beautiful course. Actually, I audited and then I didn't finish because by the third week, we were just doing 4 vectors for special relativity and all kinds of things that were so far removed from anything I would ever use that, you know, spending 25 hours on the homework assignments just was no longer a valuable time investment. And I think one of the luxuries of graduate school is you can kind of take the courses that are really helpful, and you have all these departments that offer all these courses that you want. And I really think if a course doesn't help you, then just don't take it. I mean, that's really at the end of the day.
Yeah, I think that's really true. I've dropped a couple courses that I've taken and just like learned what I wanted to know from the class by the drop deadline, or like I didn't think it was that useful. But the classes I've taken here have been really good. I've also taken, actually, I've taken quite a few classes. A lot of them are like tangentially related to my research. So I took my first semester here, I took a medical entomology class that talked about how diseases are transferred from insects. Um, I've taken a lot of fun classes. Right now, I'm in a rock climbing class. So we offer that for grad students too, which is always a good break from everything. But I've also taken classes like immunology, DNA biology, kind of the classic things that you might need to know for more advanced research. At this point, does anyone have any questions? Oh, Nicole wants to jump in.
So
go ahead.
I don't know if I know when I was looking at grad schools, like initially, this was not something I was paying close attention to, but Cornell is pretty great because they have a low course requirement, I guess. Like they have like 1 or 2 courses maybe that you have to take. And then everything else is whatever your committee thinks you need. So if you have a lot of experience, like in something, I've taken all the courses in undergrad, you might not have to take many courses at all. If there's something you want to learn more about, you're free to do so. But it's really like up to you versus, I know, like not bashing Hopkins, but like at Hopkins, we had to take a ton of courses. Like it took 2 full years of like just courses. So slight difference. Yeah. So look at what your PhD programs are offering. But yeah, Cornell's pretty good in that respect, I think.
Yeah, I think you take, so I don't remember what the required courses are, but one of them is core concepts of disease, I think. And then you have to take like 6 semesters of seminar, which is like 10 hours per semester, which isn't bad. So it's really not that bad. We have a question. What is the maximum course per semester?
I think as many as you can take, as many as you're willing to
As
burden
many as
yourself.
you can. Yeah, as many as you can take while still doing appropriate research. So it's really a conversation with your advisor most of the time. I think I came in to grad school really excited to take all these new courses, and I did take a lot of really good courses, but you get very tired of it eventually and you just want to stick to research. So I think that question will change slightly as you get further in the PhD program where you're not asking what is the maximum, you're asking like, what is the minimum amount of coursework so I can focus on research, because research is kind of the main thing. Any other questions? I think we have 5 more minutes. Okay, any other fun stories from Nicole or Thomas about Cornell, about your time here?
Ithaca is beautiful. Just throwing that out there. I know you probably see all the Ithaca's gorgeous things on like the website that people are putting together, but it is a really pretty place to live. Like all 4 seasons. I'm from Southern California, so we can get a tour of the seasons from Suzanne.
So if you do instrumentation, you also have the option of going to New York City and doing your research there. And I thought about doing that because something seemed interesting. But especially with COVID and being locked up in New York City, I mean, there are so many reasons why I'm so happy that I stayed here, basically. I think being locked up in New York City just doesn't sound attractive by comparison to being here. It's really wonderful.
Suzanne is switching through every background of Cornell she has. They're all amazing pictures.
Like,
yeah, it's kind of hard to
you
see.
Like as you're walking like on campus, like that's like a view that you get. Right?
Yeah. It's kind of hard. I'm sure it's very hard virtual format to like really understand what a place is like, but there's lots of people who enjoy the outdoors here. Although there are definitely people that I know who do not like it at all. So I think this can cater to both. We have a nice downtown area, a lot of good waterfalls. Suzanne still has more pictures. Wow.
I'm trying
that
to be too disruptive.
No, it's really nice. We're right on Cayuga Lake, so I love to swim. And so in the summer, it's amazing. You can go kayaking, paddleboarding, boating if you have a boat, I guess. But that's a really nice area too. Okay, any final questions from anyone? Do you guys have labs that you've picked out already, or are you just exploring your options?
You can do like a thumbs up if you know a lab.
Thumbs up if you know, if you have it narrowed down to like 2 or 3 labs that you're interested in. No one knows. Does anyone have it narrowed down to like 1 lab that they're going to join?
I think I'm kind of interested in the Yadon lab. I'm looking at, I have about 4 different options, probably based on what I saw on the internet. So the Yadon lab, the James Butcher, if I'm correct, if that's the name.
Jonathan Butcher, yeah.
Yeah, then the Antaki lab, yeah, and the Fishback lab. Basically, I chose them because they deal more with cardiovascular disease. So I think it's something I like to do. And just looking at how to, apply probably materials and instrumentation to solving cardiovascular disease in general. Yeah, that's for me.
Yeah, that sounds great. It's great that you have a couple labs you're looking at. That's always a good thing. It looks like people need to leave for faculty meetings. If you guys have any questions, all of the grad students are more than happy to answer them. I'm sure you have Belinda's contact. She can put you in contact with anyone. But if you would like our net IDs or our emails, we can send them to you as well. Just let us know in the chat.